Welcome back, friends. In this lecture, we shall going to touch about the centrioles and cilia and flagella. Centriole are cylindrical organelle found in the animal cell. Centrioles are small hollow cylinder that occur in pair in most of the animal cell. So centrioles, they are only present in the animal cell and they are small hollow cylinder. So its appearance is just like tuseme ni kama vile bomba, they are hollow pale ndani zina space ambayo ni tupu. So in centrosome, and centrosome we call the poorly defined structure which initiated the development of the microtubes. So it is not definitely, it cannot definitely explain, but it is made up of the centrioles. So the poorly defined structure which initiates development of microtubes during cell division, we call it as a centrosome. Plant cell, they have centrosome, but they don't have centrioles. In the animal cell, the centrosome is made up of centrioles, but in the plant cell, the centrosome is not known it's made up of what, but it initiates the development of microtubes during mitosis and during meiosis. So the two centrioles lie at a right angle to each other. Each contain a 9+3 pattern, 9+3 pattern of microtubes triplets. A ring having 9 sets of triplets with 9 in the middle. Before an animal cell divided, the centrioles replicates. Then each pair become parts of the separate centrosome. During cell division, the centrosome move apart so that each new cell has its own centrosome. Plant cells have the equivalent of centrosome, but it does not contain centriole, but absent in flowering plants. So all plant cells, they lack centrosome and they lack centriole. They have centrosome, as I told you before, but it is centrosome lack centriole. Animal cells contain a pair of centrioles in the cytoplasm, which usually lie at right angle to each other due to the nuclear membrane. So I can explain a little here. That is, if this is the cell and has only one pair of the centrioles, to say they are perpendicular to each other, during cell division they replicate. So after replication, the centrioles, they can be seen as two pairs and they move to different poles of the cell. So here we can have one pair of the centrioles and here we can have another pair. So during cell division, at the next stage again, now here the cell can divide in this way, and here we shall have one pair and here is another pair of the centrioles. So each cell end up with the one pair of the centriole. And what we can say here is that the region of the location in the cytoplasm is known as the centrosome. It contains two centrioles. During cell division, the centriole migrate to opposite poles of the cell where they synthesize the microtubules of the spinal. They are short cylinder having 9+3 pattern of microtubules arranged such that they contain group of microtubules, each of which has 3 microtubules. So here the diagram is not seen, but it is 3 microtubules, and these 3 microtubules, they have 9 pairs. So say this is the first pair, here the second pair of the 3, here is another 3 microtubules, here we may have another 3, here we may have another 3, here maybe we may have another 3, here we may have another 3. So it is 1, 2, 3, 4, 5, 6, 7. Here may have another 3, and here may have another 3. So they are 9. So what happens here, these pairs of the microtube, they join to each other they join to each other like this. They join to each other like this. And after joining, now they are forming the structure appearing like this. So this is the structure of the centriole. They are forming the structure. And here downward, there are 3 pairs again of the centriole. So that's why we call it the 9 plus 3. That is, we have 3 pairs of the microtubules and they are 9. So this is just one centriole and it is like side view. So as I told you that in the cell they appear, or their position is perpendicular to each other. So if the position of one centriole is like this, then the position of the another centriole, it will be like this, and here the angle will be 90 degrees. So function of the centriole. They are used in the formation of spinal apparatus during cell division. They are concerned with the formation of cilia and the flagella, and they are responsible for the production of the microtube. So the microtube produced can be assembled to form the basal body of the cilia and the flagella. Now let's see a little about the cilia and the flagella. Cilia and the flagella are organelles that have identical structure, although flagella are longer in structure. Their structure used for locomotion. So flagella, they are longer, cilia, they are short, but in structure they are similar. In eukaryotic organism, they are complex and hence microtubes arranged of 9 plus 2 pattern and do not have, and they have basal bodies. So here, when we are saying the 9 plus 2 microtube organization, I think I explained it in the previous sections, but if you would not be together with me, I can draw to you here. So let's say this is our cilia, and here we have the inner membrane of our cilia. And now, when we are saying about the 9 plus 2 microtube organization, let's say we have 9 microtubes outside here. So it is 1, 2, 3, then it is 4, then it is 5, then it is 6, it is 7, it is 9, it is 8, and then it is 9. So it is 1, 2, 3, 4, 5, 6, 7, 8, 9. And then here at the center we have 2 microtubes. So that is what we call the 9 plus 2 microtube organization. 9 plus 2 microtube organization in the cilia and the flagella. And this is found only in the eukaryotic cell, but the prokaryotic cell, they don't have this. So function of cilia and the flagella. Flagella and cilia are locomotory structure. They are used for locomotion. The second thing, in the respiratory tract, cilia are used to trap dust particles taken together with inhaled air. The third thing, in the urinary bladder tubules, cilia contain flow urine in one direction. They are involved in the flow in the urine in one direction. The fourth thing, in the oviduct, that is the fallopian tube, cilia, they are involved in the movement of the ovum, used to transport the egg from the ovum, from the ovary into the uterus. The fifth thing, in the brain ventricles, cilia contain the movement of the vertebral spinal fluid. So the cerebral, this is not vertebral, it is cerebral spinal fluid. So cilia, they are involved in the movement of the cerebral spinal fluid in the brain ventricles. Point sita, the energy is also used to acquire food. E.g., feeding current generated by the paramecium. So the cilia can be used during movement, and that movement enable the organism to acquire food. The seventh thing, they are used to sense environment. They act as sensory hairs. And here we have few differences between the cilia and the flagella. So cilia, they are short, flagella, they are long. Cilia, they are numerous, but flagella, they are few. And normally, you can see a cell with only one flagella, but you cannot see a cell with one cilia. So let us just finish with this part. We have the non-living part of the cell, and the non-living part of the cell, they are called cytoplasmic inclusions. So they are non-living structures of the cytoplasm. They are also known as ergastic bodies. So example of non-living part or cytoplasmic inclusion structure in the cytoplasm, they are food granules and crystals of various substances, mainly waste products of metabolism, such as calcium oxalate, which are found in citrus fruits, calcium carbonate, and silica. So the food granules are such as glycogen in animal cell and fungi, starch in plant chlorophyll, green algae, and euglena. So on the concept of cell differentiation, we already discussed it in the beginning of this cell. We already discussed the concept of cell differentiation. So let's touch a little on the concept of cell potence as we are going to finish this topic. Cell potence or cell power is defined as the term which describes a stem cell ability to differentiate to cell types. So stem cell, they have ability to differentiate into different cell types, and this is done especially in the not only in the plant, even in the animal cell. However, in plant it is different from the animals. In the animals, if you take the stem cell means there are cells which can differentiate into different cells. So in animal, when you are taking one animal cell, and then after taking that one animal cell, you can just enable it, giving it environment for it to replicate. But in the plant, even the adult cell can differentiate into different cell types, as we shall study in the totipotence experiment during genetics. And I hope I will lecture it. So significance of the cell differentiation. And I don't want to discuss these because we have discussed them during the topic of cell differentiation. So we have different questions on the cytology, and we have different concepts. But I can tell you that this is the end of cytology. I wish you nice studies. Next topic, according to our Form 5 syllabus, is biochemistry. And after this, I will jump to biochemistry. So wish you nice studies, and we shall meet in the biochemistry lectures. We have different concepts. If you have any question on cytology, just comment my videos. If you haven't understand anything, comment my videos, and I will send the question in my Telegram group. Just click the link below in the description to join my Telegram group, and then you will see the questions there of cytology. And even some of the papers you have already sent there, they contain some of the questions of cytology, and I'm sure they are enough for you to practice and to gain more and more knowledge on cytology. Wish you nice studies. This is the end of cytology. Let's meet in biochemistry. Thank you.